يعني مو شرط انا ما كنت فاني حبيبي
Okay. All right. Let's start. So in the last lecture, we talked about remember independent assortment on the last slide. I think it was the last slide. So if you're considering two traits, remember the dihybrid cross, and if they're located on different chromosomes, they can arrange independently of each other, those two traits, okay? So,
I'm sorry.
you have your coffee this morning?
No.
No problem. Okay. So this applies, this law of independent assortment only applies to non-linked alleles. What that means that they're not on the same chromosome, okay? So if alleles or traits or genes are linked, they will be located on the same chromosome. Then this independent assortment does not apply. Do you remember from the lectures earlier in cell division that we talked about that? Independent assortment. What is it? I'm listening.
Crossover.
Crossover?
Can you repeat the question?
What is independent assortment? Anybody draw it for me?
Yeah.
Yeah.
The Mendel did it.
Yeah, Mendel did it. But
Oh, أول حاجة.
so
This is
Green
when
and,
the egg and
yeah.
sperm.
yes. So during meiosis 1, independent assortment happens during meiosis 1. So you have the duplicated chromosome here. So it's carrying. What am I doing? It's right here. Okay. Have a look at here in this combination. The red on the left side you have one red,
One blue.
one blue.
Yeah.
Right side one blue, one red.
Yeah.
But in another combination both red or blue.
This is in.
So they can, as these two can assort independently of the other pair, okay? So each pair of homologous chromosomes can assort independently of one another. And if we consider we have 23 chromosomes, okay? So what is it? 2 to the power of 23, which was like about 8 million different combinations we can get from this, okay?
Can you show us an example for linked
Yeah, I'm gonna go into that one next.
Hasn't it?
So that's law of independent assortment. Now this doesn't apply for linked alleles, okay? Now linked alleles, if you have a look at the chromosome, they're on the same chromosome, okay? For two different traits, say for straight or widow's peak, straight hairline widow's peak, or attached and unattached earlobes.
The same chromosome.
They're not on the same one, but I'm just giving an example, okay? As a trait. So these two will be inherited together because they're linked, they're on the same chromosome. Unless crossing over can, separate them, okay? So if you have crossing over and they can be separated to the non-sister chromatid, okay? On the second pair. And that depends on how close they are, okay? So if the alleles are very close, then they might be, during crossover, both of them might be carried to the non-sister chromatid. But if they're far apart, okay, say one on the...
They may separate.
They may separate, okay? Depending on how they are, okay? Located on the same chromosome.
Okay.
If they're close together, they're not the law of independent assortment doesn't apply to them, okay? They're not sorted independently of each other, these two traits. Yes?
So if the crossing over happens, it won't inherit it together.
It might not, okay?
So there will be a chance of not being inherited together.
Yes.
What does it mean to be inherited together?
So we're talking about the law of independent assortment, remember?
No,
The dihybrid cross.
I don't.
So, were you here last week?
Yeah, I was.
You was here? Okay. So you remember here in the dihybrid cross, we write the gametes with the different traits, different alleles, okay?
Yeah.
Now during meiosis, those they separate, okay?
Yeah, I'm.
Each allele separates if they're located on different chromosomes, okay? So if they're located on the same chromosome, they're not going to separate. They'll stay together. They're linked.
Yeah.
So this law of independent assortment is established based on the hybrid cross that Mendel did, okay? And the traits are located on different chromosomes,
Yeah.
okay? If they're linked, so that means that these traits that the law of independent assortment applies to are non-linked, okay? For linked ones, okay, they may be inherited together, okay, if they're close enough. So
They get close.
Yeah.
They will.
sometime crossing over can reshuffle it, okay,
And then they will
Move.
be not close
Yeah,
enough to.
yeah. move it to a different,
Chromatid.
the non-sister chromatid, and they might not be inherited together, okay.
Oh.
So, and that depends on how far they are, okay, from each other. So if they're close to each other, even during crossing over, they can be inherited together.
Is there a certain length that
Uh,
has been done?
No, I mean, we just
Depends on the
Basically.
crossover.
crossing over here.
Yeah.
So this is a random thing that's happening. So if close enough, they are,
Inherited.
the higher the chance they'll be inherited together. The more far apart they are, if crossing over happens, the less chance. And remember we talked about this genetic variability before in the cell division. So crossing over was one of the ways you can get genetic variation. Another one is this independent assortment that we talked about, okay? So independent assortment, we can get how many different combinations? 2 to the 23, so about 8 million or so, more than 8 million, okay? So we got independent assortment, we got giving us about 8 million, reshuffling of the chromosomes, okay? So crossing over, giving an increased variability, and then you have the random chance of an egg being fertilized by these
Sperm.
300 million sperm. So the variability, these all contribute to the genetic variability, all these processes.
One process happen or all of the three?
All of them.
So independent assortment is happening, okay? In meiosis. Reshuffling is happening as well, but depending on where it's happening, okay? This is going to be more random. Both of these are random, obviously. Well, all of them
All
are
of them are
random.
random.
Yeah. So,
I think the last one is certainly.
so out of those 300 million sperm that were produced by this process, one of them,
We're fertilized.
yeah. So if you do the statistics, the probability, you have to multiply the chances of each one together, and it'll be a lot. I don't know what it is. Okay. The first one is 8 million. Partial reshuffling might cause
We don't know.
another million. I don't know.
We don't
And
know.
then you got 300 million here. So you multiply it.
Big number.
It's going to be a big number. That's why we get lots of genetic variation. Okay.
Have many sons, you see, genes.
Well, you'll see them in there, but they're just reshuffled. Okay. So linked alleles may be or may not be inherited together. Okay.
What I said in the lab.
All right. So let's talk about some non-Mendelian genetics. Okay, where there is the recessive or the dominant is not fully expressed. Okay. So one case is the incomplete dominance. Okay. So there is an intermediate phenotype between the two dominant phenotypes. Okay. So if you find the alleles, for example, for Caucasian hair, so in the white you find if you're homozygous for the straight, you get straight hairline. Okay. If you're homozygous for the curly hairline, so this is the straight hairline like that. Homozygous. And if you're homozygous for the curly hairline, you'll get curly hairline. Okay. When you cross those two, so what you get is all the offspring here will have this wavy hairline, which is an intermediate between the straight and curly. None of the alleles are dominant over each other. You get incomplete dominance, a phenotype in between the two. Okay.
This is only happen with the hair.
There is some more examples we will show as well. Okay.
It's also applied in the color in the hair, right?
Yeah, it applies in.
اسمع
So remember, in color
So
of
now
the
it's
skin,
not dominant, not
من كان هاي بعد
it's incomplete dominance.
شكلك انت عارف.
So both of these are by themselves. Okay, when they are homozygous form, they show the phenotype, so straight or curly. But when you cross the two, the heterozygotes, okay, the heterozygous is not dominant of each other. It is an incomplete dominance where you get an intermediate between the two. Yes.
What about the hair color?
So in hair color, skin color, these are polygenic traits. Okay.
And this is just
Eye color, height. Okay. Many genes control them.
this is just with the heterozygous.
Yes. So if you look at the heterozygous of those you will also find incomplete dominance. Okay. So you will have if you cross a very tall person with a short person, you don't get short or tall. You get medium. So it is incomplete dominance. Okay. So a homozygous tall person crossed with a homozygous short person will give a heterozygous medium, incomplete dominance. Okay.
Children get taller when their fathers.
Maybe they're carrying the.
Grandfather.
Yeah.
Alright.
So if you have two heterozygotes cross them, if some of them will come out
Or maybe it's related to the
a
environment.
homozygous phenotype.
There are chance for this happening. There are some percentage, some scientific.
I don't know the percentage, but yeah.
Yeah, there are some.
Doctor, dark color is dominant over the light color, right? For the skin.
Yeah. All right. Let's. Okay. So we understand that in the homozygous form, each allele is dominant. Okay. Well, it shows obviously homozygous. It shows either straight or curly. But when you cross the two, you don't get. None of them are dominant over each other. We call this incomplete dominance. You get an intermediate between the two. So in this case.
Could it be straight but CS?
No. Okay. So if we let's do another cross.
So doctor, if the parents are both.
If the parents are both
No, I don't mean
like that, huh?
No, I don't mean that. I mean,
SS first.
if there is.
SC. So there is a chance, yes, to get.
No, I don't talk about this. If the both parents are straight, is
Yeah.
there any chance to be curly or wavy?
No.
Yeah, there are chances if you from
Your
grandfathers or.
There's no chance, because if you're straight, that means you're homozygous. You must carry this.
Oh, no. I saw a lot of parents with a specific trait and their children.
Yeah, but this is different. Okay, we're talking about the hair.
What we talk about?
Yeah, I'm talking about the hair.
Kala, what are you talking about? Hair shape?
Yeah, hair some changes.
What do we
Maybe
do?
some two parents are very white and the child become brown.
No, we're not talking about that. We're talking about this. Okay. What,
Only the hair.
yeah.
The hair,
The
it's
color
not kind
depends
We're
on-
talking about specific phenotype here. Why are you going to something else?
Okay.
But we do this for all the traits.
No. سيء.
No.
ال-
I'm
color,
talking about
skin
this
color,
trait
hair
here
color.
only. Okay.
أشياء ثانية تأثر عليها لا.
So, Doctor, the both parents are
dominant
dominant.
بهذا الشكل.
So
لأنه يأثر على أشياء
we
وايد.
get incomplete. Well, in the recessive form, they will show their phenotype. But in the heterozygous, remember, in the complete dominance, one of them will be dominant, the other one will be recessive. In this case, in incomplete, you get expression of both, causing an intermediate. Instead of being straight or curly, you get in between, a wavy hairline. Okay? So it's neither of them are dominant. Okay. They're not dominant or recessive, but there's incomplete. You see a intermediate between the
They
two.
consider both of them are complete. Or both of them are recessive.
Well, in the heterozygous,
No,
we can't say they're dominant here, okay? Because there's
There's
homozygous.
no one stronger than the other.
Yeah. It's all right.
Well, so doctor, this is just in this phenotype and why? What's the difference about the shape of the hair?
One is curly and one is straight.
Okay. Why it's just this in this phenotype? If your father are recessive and your mother are recessive, you take this one intermediate.
Are you talking? There's no recessive here, okay?
It's both of them dominant.
Okay. So what we're seeing here, in the homozygous form, each allele will have its own. It will show its phenotype, okay? But in the heterozygote form, okay, this is where it's incomplete. Here it's not, okay? The incomplete dominance is showing in the heterozygote, okay?
Okay, but I asked about why this phenotype. It should be both of the both of the father dominant. The son cannot have straight hair maybe or wavy hair. Both of them straight hair.
Both of them straight hair because they're both carrying the S.
Yeah, but in another like the skin color, there is some
We're not
chance.
That's
why this phenotype like
That is a polygenic trait, okay? You know what a polygenic means? Many alleles.
polygenic.
Yeah.
Doctor, is there any chance for a mutation to affect the hairline shape?
Well, I mean, of course, there will be some mutation. If you get a mutation in one of the genes, that-
Will affect.
Yeah, of course, any mutation, depending on how severe it is. Okay. This is a single. Okay.
So the mutation أثر بعد يمكن يكون في mutation.
All right. Let's move on to codominance. Okay.
بس أنا وش مميز الفينوتايب هذا اللي يصير كده هو؟
ما أعرف ليش هو الحال ما أعرف. قال في مشكلة.
So we see codominance in the blood type, in the AB blood type. Okay. You remember that we did this in the lab, huh? You done that in the lab, the blood test?
Yeah.
So you did that with the synthetic blood. So the blood typing is controlled by 3 alleles, okay? ABO typing, okay? So if you have blood type A, that means you're on the surface of your red blood cells, you're carrying the A antigen. So polysaccharide A. So it's a polysaccharide, the antigen, okay?
The antigen polysaccharide.
Alright? So that antigen A is polysaccharide A. It's on the surface of the red blood cells. For type A. For type B, it's polysaccharide B. And it's on the surface of the present on the surface of the blood cells if you have type B, okay? If you have AB, and this is where the codominance comes, okay?
You have both A and B.
You express A, polysaccharide A and B equally, okay? So 50-50. 50-50 ratio of A and B on the surface of the red blood cells. This is codominance. It means that they're both equally expressed, okay? So codominance is with both of them are equally expressed. And if you don't have any of the polysaccharides, obviously you will end up with O, okay? That means you have no antigens, okay? You don't have the I or B antigen on the surface. Your blood type will be O.
Doctor,
Yes.
how does codominance happen?
Codominance happens when both alleles are expressed
Expressed
equally.
But when do we know that it's expressed equally and when?
So in this case, in the blood type, we know, if you're AB, you express both antigen I and antigen B on the surface of the blood cells equally. Okay, that is codominance. Or if you see, sometimes you see it in animal colors. Okay, or
So there are certain situations where we can.
actually that's in complete dominance. Yeah.
There are certain situations where it's codominance in some genes.
Yeah, there is. So we'll talk about the sickle cell anemia next. I'll show you that case.
Yeah.
Okay.
Inheritance of
Yeah. So there is another case where you can see in the disease codominance as well. All right. So as I mentioned again, you know how, the genotype. If you have two A alleles, obviously
You're right.
A, and if you have one A allele and one
No allele.
no, so you got one A allele from your say mother and no allele from your father, you're AO, okay?
So your group blood it's A.
So yeah, this is complete dominance, okay? In this case, all right? Because remember, remember that how we write the blood type, capital I A, capital I A here as well, okay? For this one, capital I A, small I, okay? There's three different alleles, remember?
Small I it's mean without
Without antigens.
without antigen.
The three alleles that control blood type, okay? So in this case, you can have
IB,
small I, okay? In this case, so this is where you see the complete dominance. Both of the alleles, both of the antigen A and B are expressed equally, okay? And the O is recessive. To get this phenotype, you must have none of the antigens, okay? So
What's the
B and O, B, A and B are dominant over O. So
B and A?
yes, A and B are dominant over O, okay? But when you get A and B together, you get codominance, equal expression of both antigens on the surface. Yes. So this is codominance.
Doctor, what's the organ responsible for produce this antigen?
Well, it's produced by the red blood cells. You got the hematopoietic stem cells that originate the red blood cells. So red blood cells don't have a nuclear, remember? So during the development, they will acquire these antigens on the surface because these antigens, they will have genes that are expressed in the DNA. So red blood cells are made in the bone marrow.
So, what's the special about the red blood cell that does not produce antigen? It's different from what's
Well,
produced
you don't have the
in
gene, you don't have the allele, it's genetics.
Okay.
What are we studying here? Genetics?
So it's all about your father and mother.
We're talking about the DNA. Genetics is all to do with your DNA. So if you don't have the DNA for that, if you're missing the allele, the gene for the antigen
But there
A,
may be a mutation. If your father A and your mother B and you'll be O.
if there's
It's-
a mutation that
it's
causes,
genetic.
I'm not sure of any diseases.
It's
that
not
have-
disease. I think it's different. If you A and
Well-
B and you'll be O.
Yeah. So I'm not aware of the mutations. Okay. But a mutation can affect. If there is a mutation in any gene, it can affect its expression, depending on how severe the mutation is, how it disrupts it. Okay.
Is there another part?
Doctor, by polysaccharides, we mean antigens.
Yeah, the antigen. Yeah.
They are the same.
Yeah. Okay. All right. Here's a little activity here. It's very easy.
Yeah.
So you got a male that's IV, a male that's A, and a female that's B. They have a child with O. Come here and solve this for me. What you gotta find the missing parents of this child is misplaced. The parents is one of those. So what about this one? What's the alleles for that? Yeah, but if you didn't, we should do it to show the working. Show us the working why it's not the answer.
So this is impossible.
Because it will be A B A B. This is B. This is
They are not his parents.
because
It's impossible.
He's
because
adopted.
B and A is dominant.
He's adopted.
So that one is impossible.
He has not his parents. He's adopted.
He's adopted. Okay.
ويا الاي ويا البي الحين بيطلع بالزاوية اليمين الميل اي ويا الفيميل بي
So what's the phenotypic ratio for that? For the O. 1 out of 3, a quarter.
Oh,
Yeah.
the
One
ratio.
quarter. Yeah. 1/4 of the children. 1 out of 4. So yeah, 1 out of 4. Yes, that's right. Now we did the working out to show that it can't be male AB. But remember, you can easily drive to the answer if you know that O is recessive. Okay? if this male AB, okay, his allele, he doesn't carry one of the recessive alleles, okay?
So it's impossible.
So impossible for him to contribute to the
Oh.
O. So immediately no, it's not that. Yes.
If the male is AA, but and the female IA, IB, and IB.
Then it's impossible, okay?
But it's.
So in this case, the male has to be, it has to be this one, okay? You have three choices, okay?
But it doesn't say that. If it is
Well,
IA
we
or
say,
IA.
this question says one of
بس ديك
the
impossible
parents
تصير.
are in these
مستحيل
three.
ديك تصير.
بس ما ذكر في السؤال اذا الجيل الثاني حقها او ولا
اي مو قال لك انزين بس خلاص انت عارفها لان ديك impossible.
بس اذا تصير ولا واحد منهم.
Read the
ما
question.
يصير ما يصير.
Read the question. This is a child that belongs to a couple from the above. So it belongs to one of them. What does that mean? That two of those three are the parents. So we assume that if they're the parents, this one
Should be IA.
cannot be in this form, okay?
Yeah.
Because then it will be impossible to have
Any child.
that child with O group, okay? And the same applies to the female as well, okay? So she cannot be,
BB.
both of them must be in that form. They must be in the heterozygous form, okay? For B. They cannot be homozygous dominant for B. You got that?
ايوه ماما.
So we answered that question.
ايش فيك هذا؟
All right. So as I mentioned, another example where we see this non-Mendelian genetic inheritance is sickle cell anemia, which is very common in this region, isn't it?
Yes.
And it's common in the trite, the sickle cell trite is very common in the African region because it actually people with the sickle cell trite are resistant to malaria. So sickle cell trite is resistant to malaria.
Impossible to have malaria
Yeah.
and.
Yeah. People with sickle cell trite cannot get infected with malaria. Okay? So that is an advantage for those people in Africa, okay? All right. So if you have a look at the sickle cell trite, remember that in the red blood cells, the protein that carries the oxygen, hemoglobin, okay? We talked this a little bit, we did that activity last week, I think it was, or the week before on the sickle cell anemia, remember?
Yeah.
So the HB gene, the hemoglobin gene, it has a mutation in it, okay? Just the point mutation that causes this sickle cell anemia. So if we have a look at this, the normal hemoglobin is denoted as HbAA, okay? The normal allele, alright? The sickle cell allele is denoted as HbAS, okay? So normal is A, HbAA, or if you have the mutation in the allele,
Yes.
the allele is denoted as HbS. And to get the sickle cell anemia, both of the alleles must be sickle cell, okay? If you have one normal and one sickle cell,
So
you're a
the,
carrier, you're normal,
so the normal is dominant.
normal depending on conditions, okay? Sometimes some of the cells can go sickle if you're in prolonged exposure to low oxygen, can cause some of the cells to go sickle cell.
Even if one of it's
Yeah,
normal,
even in the carrier.
even in the carrier.
Yes. But normally they're okay, okay? So it's not-
Normally it's dominant.
they're normal. The normal hemoglobin is dominant over,
The sick.
and this is where I'll talk about another case of incomplete dominance in this year. Okay, like all right.
This is another example.
Yes. So normal is carrying both HbA. The heterozygote is normal, okay? Unless there is low oxygen, and then the cells, some of the cells can go sickle cell, all right?
Even if it can.
Yes. So normal is dominant over the, in terms of the shape, okay? In terms of shape. But we will see here that we see codominance in terms of expression of the protein. So let me go here. So we see both codominance and incomplete dominance in sickle cell anemia, okay? So normal, if you to get sickle cell anemia, it's a recessive, okay? So you must have both the HbAs alleles. So sickle cell anemia is homozygous, okay? The gene involved in that is, remember, the HB, I think from the last one, what was it? HBA gene. It has a mutation in the 6th amino acid. Remember from the last activity we did on the sickle cell anemia?
Yes.
So it has a mutation, okay? I think it's the beta hemoglobin. Beta hemoglobin, I think it is. It has a mutation in the 6th amino acid. So that's what's causing this mutated form of the protein to be formed. Now, what happens in sickle cell anemia is that in low oxygen, this mutated form of the hemoglobin crystallizes, okay? And this causes this crystallization of the hemoglobin causes this sickle cell, okay? So to get the phenotype, the sickle cell anemia, you must be carrying both HB, so you have to be HBS, okay?
Homozygous.
And you will get the sickle cell anemia. The heterozygous form of it, okay? This is where you see, where we can see the codominance and the incomplete dominance, okay? In terms of codominance, what we see is that both HbA and HbS are roughly expressed at 50-50%, okay? Both hemoglobins, okay? The normal and the mutated hemoglobin are 50-50. So this is where we see when they have both hemoglobin, okay? At 50-50. So this is, in this case, when both are expressed equally, this is codominance, okay? In terms of the expression of the protein. But in terms of the shape of the cells, we see incomplete dominance, okay? Normally, it's dominant, okay, in normal oxygen conditions, okay? The HbA is dominant, okay?
Doctor?
The shape is normal, okay?
Yeah. But
When you get low
the oxygen.
oxygen, some of the cells starts to form sickle
So-
cell and some are normal. So it's incomplete, okay? The shape, in terms of shape, we see incomplete dominance,
So
okay?
the function
mainly
will
caused
be
by
also
Yes, function will be reduced, okay? In low oxygen, okay? It happens in low oxygen, okay? Very prolonged exposure. But normally in these conditions, the shape of the red blood cell doesn't change, okay? There is some exceptions, but we don't need to know about that. in the kidneys, in the cortex, where there's hypertonicity, that it can cause some sickle celling of the red blood cells. But normally, overall, you see normal shape, okay? The donut shape. So normally you see that shape, okay? If the oxygen levels drop, some of the cells will sickle, okay? And this is where it's incomplete dominance. So HbA is no longer completely dominant, okay? So you get some sickle cells forming, and the trait becomes incomplete dominant.
So in the first circumstance, it's complete dominant.
It's co-dominance. At the level of protein, both are expressed equally. Both the normal and the S, HbA and HbAs are expressed 50-50.
But it's normal.
Yeah, we're talking about the protein.
Okay.
The shape is a different thing, okay?
Okay.
So in terms of protein, it's co-dominance, okay?
But in the shape, it's
In
not.
terms of the shape, we see complete dominance in normal conditions, okay? And incomplete dominance in low oxygen, okay? Is it clear? Right there in the back there. In what sort of condition would we see incomplete dominance in the sickle cell trait?
Less of oxygen.
In what condition in the sickle cell trait do we see incomplete dominance? In terms of the shape of the cell. Do you know?
Can you repeat it again, please?
In what condition would we see incomplete dominance in terms of shape in the carrier? The heterozygous. Low oxygen. It's written there. Right here. Okay? So we don't see the trait for the shape of the cell is dominant, okay? Normally. But if the cells are exposed to longer periods of oxygen deprivation, okay, some of the cells will form these sickle cells, okay? They will form the sickle shape. And people with this, which are carriers, they don't have many symptoms normally, okay? They don't have the severe symptoms of the people who are homozygous, who have sickle cell anemia,
What is the symptoms?
You can get multi-organ damage, okay, because of oxygen deprivation. The cells cannot efficiently carry oxygen.
I think if you start too fast, some get
Yeah. These people also, they can't put too much, they exercise.
Yeah, not too much effort.
Not too much effort into things because their red blood cells are already struggling to transport oxygen, and if you put more demand for oxygen, it's gonna make it even worse. Okay?
So the guys, the low oxygen is both of them, the shape and incomplete.
So incomplete dominance is in terms of shape, okay? We see complete dominance is in incomplete, depending on the condition. So this is an example where the environment plays a role. Remember, we said there's genetic and environmental factors.
Yes.
So the environment can play a role
So
on
the
the.
environment effect on
Yeah.
living thing.
Well, it's the oxygen. So oxygen is from the environment,
So
okay?
in general,
If you go to low oxygen, you will get some of your cells just form sickle cell. So not the genetics is not all at play. The environment also plays a role.
so in general, the environment affect enough.
Yes, yes,
The living thing.
yes. The environment and your genotype together work to give you a phenotype. In this case, you can see that the genotype and the environment working together to give that incomplete dominance.
Doctor, so I'm a bit confused
Yes,
in just two terms.
yes.
So what's the difference between codominance and incomplete dominance?
Codominance is when you have a 50-50
Yeah, okay.
equal expression, like AB blood type,
Okay.
like HbA and HbS expression at 50-50 roughly.
Alright, teacher.
Incomplete is when you get an intermediate, okay? It's not completely dominant, okay?
Okay.
Each one of the alleles is not completely, you get in a between, okay?
It's heterozygous.
Yeah. So in this case, HbA in normal condition is dominant, for it causes a normal red blood cell phenotype, okay?
Okay.
The shape of it.
Yeah.
Complete dominant.
If the oxygen levels are dropped to low, very low,
Mhm.
some of those cells will start to form the sickle shape.
Sickle cell.
So this is incomplete dominance, okay? This heterozygote is incompletely dominant.
Oh, okay, I get it.
In terms of shape,
Okay.
yes.
And the homozygous sickle cell anemia, the people who have it, they usually get the scissor, when they, yeah, the seizure.
Maybe because of low oxygen in the brain.
The carrier got this situation. They
Now
can
the
get
carrier is
seizure.
normal, no symptoms, very few symptoms.
Only
Yeah,
sometimes.
there is no big symptoms.
No, carrier is fairly normal. Unless you go to, I said, low oxygen, where it can affect the cell shape. And that's when you get
Or you will
incomplete.
say some pain or something.
No, I think this one is normal, there's no symptoms.
Yeah. So it's only
It's just the
turns
carrier.
to a sickle shape,
There is
but
no,
there's pain or something.
they don't do any, there's no need for any intervention in this one.
Yeah.
It's normal. Yeah. But that people have to be careful of with this sort of trait, not to go in like hiking in the Mount Everest or something with low oxygen. It might.
I think the color of it's very cold. People who I know some people they turn
Bluish
bluish, yes.
because of low oxygen.
Yeah, purple.
In the, yeah. That means there's less oxygen in the tissue. 1 minute.
This is the name of not to be confused.
We'll get on to it and then we'll talk about it again.
We had 3 slides.
We'll introduce it. Okay,
Guys,
so we know about complete, incomplete dominance, codominance, and we know examples of each one of those, okay? The next type of inheritance, which Mendel didn't establish, this is modern genetics, which we call polygenic inheritance, where more than one gene or more than one allele is involved in the inheritance of these traits, like we said, eye color, skin color, height, size, shape. So these sort of traits, they can be controlled by more than one gene working together. And working together, we get variation in the phenotype, okay? We get a variation in the phenotype. For each of the gene, they have an allelic pair. Remember, you get one from your mother, one from your father. So many genes working together, each in allelic pairs. Okay, and you get this continuous variation. So the combination of the different genes, the alleles from those different genes combined together. So in the homozygous form, each at the extreme end, so you got the dark, really dark skin. In the homozygous form, it's dominant. Okay. So it shows the dark. Okay, and the homozygous for the white skin color. So it's not recessive. It's like that. What earlier before we did the curly and the straight hair, we see incomplete dominance here. Okay, so if you cross a white person with a dark color, you don't get. You get in between here somewhere. Okay, so if you have a look at here. this is actually for height, okay? So for height, very tall people, okay? They have the capital A, capital B, capital C in the homozygous form. Yeah. I think we'll continue from this next time, alright? Because we're out of time. Okay?